Hello friends, once again welcome to new session of Hospital and Clinical Pharmacy. Myself is Malvi Rajendra Bhavsar, M.Pharm in Pharmaceutics. So today we are going to start with new chapter, that is Bioavailability. So after watching this, student will be able to learn concept of bioability, factors affecting bioability, pharmacokinetics and pharmacodynamics, factors of that which affect the bioavailability of drug. So, introduction. A drug can exert a pharmacological effect when it reaches the site of action in an active form. Now, bioavailability may be defined as the rate at which and extent to which the drug reaches systemic circulation in active form. The rate at which drug enters the circulation controls the onset, intensity and sometimes duration of pharmacological effect. Definition of bioability. Bioability is defined differently by different individuals. Now, to illustrate the National Formulary 1970 defined bioability as the extent to which active ingredient in a drug product is taken up by the body in the form of which it is physiologically active. FDA defines bioavailability as a degree to which the drug is absorbed from the drug product into body or to the site of action. It is, however, difficult to measure the availability at a site of action. Indirectly, it is measured by the pharmacological effect. Then next definition is by Community of Academy of Pharmaceutical Science. It defines bioavailability as a term to indicate the measurement of both the relative amount of a drug administered drug that reaches the general circulation and the rate at which this occurs. This definition is acceptable since it is possible to measure the compare amount absorbed and the rate of absorbed drug. Now, process of absorption. So it is important to know the manner in which drug is absorbed. The absorbed drug may be either direct or indirect, but it involves passage of drug across the cell membrane. So this passage is governed by lipid barrier present in the permeable membrane. Actual mechanism of absorption is very complex phenomena and is not yet understood thoroughly. The following mechanism occurs in the process of absorption. So first is passive diffusion. The term passive diffusion means that drug or substance is transported across the cell membrane without utilizing energy. It is also known as simple diffusion. The rate of transfer of the substance is proportional to the concentration gradient across the cell membrane. Drug molecules penetrate the cell membrane either by the way of passage through aqueous pores in the membrane or by dissolving in the membrane substance. But fat-soluble and water-soluble molecules of smaller size may cross the cell membrane by simple diffusion process. Simple diffusion is nothing but from the higher concentration towards lower concentration. So, this is what shows about passive diffusion, or you can say passive absorption, that is movement of drug molecules from the higher concentration towards the lower concentration without utilization of any energy. Then comes active transport. It is a specialized process requiring energy. This process is independent of physical property of membrane. In this process, carrier molecule combine with the drug molecule to form a complex on one side of the membrane. This complex then diffuses through membrane and dissociate into carrier and drug molecule when it reaches the other side of membrane. After this, carrier returns to the original side of membrane to repeat the process. Now, in this plasma concentration time curves are the process useful for the studying bioavailability. Differences of drug administered in different dosage form but containing same amount of the drug. This helps in the selection of a type of dosage form for the specific drug. So, this shows active transport. So, this is extracellular fluid, this is intracellular fluid. So, there is absorption with the use of energy. And here energy is used in the form of ATP. ATP is required for the transformation of particles to the extracellular and inside. After ATP, it is converting into the ADP. So ADP and ATP, this energy is required for the active transport or active mechanism of absorption. Then this is curve. From the curve, formulation A would produce quick action, onset of action but short duration of action, and formulation B produces effect of much longer time, and C therapeutically inactive because it would not achieve plasma concentration of a drug for the sufficient time. Also, the formulation A crosses the minimum toxic plasma concentration, which may result in toxicity of drug. So this is time drug graph plotted by mean plasma concentration and time in hours. So this is your minimum toxic concentration or therapeutic range or minimum, and this is minimum effective concentration. So this is graph for formulation A, this is for formulation B and formulation C. Then distribution is a movement of solute from blood into the tissue. That is, this drug will get distributed and combined with either proteins, lipid, liposomes or in the mitochondria. And blood pH, that is 7.4. Metabolism, it is a process in which chemical reaction carried out in the drug body to convert drug into compound different from the originally administered. Then excretion is a process by which drug and its metabolite are removed from the body, mainly through liver or urine. Relative viability, it is the availability of a drug from the dosage form as compared to reference standard. Absolute viability, it is the availability of a drug after intravenous administration, and bioequivalence, a product is considered bioequivalent if the rate and extent of systemic absorption dose not show a significant difference from pioneer drug product when administered the same dose of therapeutic ingredient by same route under the same experimental condition. Then factors affecting bioavailability. So rate and extent of ability from the dosage form is affected by various factors, so which are discussed here. First is physical property of drug, which includes pKa, partition coefficient and particle size. Then pharmaceutical factors include dosage form, manufacturing variable and dissolution rate. And physiological factors, effect of GIT fluid, GI transit time, first part effect and disease state. So pKa. Drugs which are weak electrolyte exist in ionized as well as non-ionized forms. So extent of ionization depend on pKa of drug, that is dissociation constant of drug, and pH of the surrounding fluid. So drugs that are more water soluble in ionized or a salt form, but ionized form is not readily absorbed. So example of organic acid which have pK values between 3 to 7 remain unionized in gastric pH fluid, that is pH 1. Thus it will be get well absorbed from the stomach. So in short, unionized drug or unionized one will absorb more as compared to ionized drug. Partition coefficient, this drug is a ratio of its solubility at equilibrium in the aqueous solution or its solubility in non-aqueous solvent. Of hydrophilic drugs have a higher water solubility and dissolution rate compared to lipophilic drug. So non-ionized form of a drug is more lipophilic than ionized form. So non-ionized form is better absorbed as a biological membrane is lipoidal in nature. So higher the partition coefficient, more will be the absorption and more will be the bioavailability. Particle size. Particle size of drug is reduced, surface area increases. So increasing surface area ultimately increases dissolution rate and also in turn availability of drug. So example, decrease in particle size increase the absorption of crystal. Then pharmaceutical dosage form related factors. So first is dosage form. The availability of drug from the dosage form is generally can be shown as solution. First, more availability will be of solution, then suspension, then powder, then capsule, tablet and then coated tablet. So product formulation factor, bioavailability decreases in the following order. Account of decrease in absorption of the following dosage form. So how bioavailability decrease? Solution to coated tablet. Solution is more, then suspension, then capsule, then tablet and then coated tablets. So first solution, they are absorbed most rapidly from GI tract. Aqueous solution are faster absorbed than the non-aqueous one. Exceptionally, however, non-aqueous emulsion also may show the faster rate. The absorption of solution is affected by viscosity, reversible complexation, solubilization and chemical stability. Then suspension. In suspension, the particle must dissolve before the absorption. That's why its solubility is lesser than the solution. Then comes your capsules. Capsule absorption problem is respect of capsule associated with diluent present with the drug in the capsule, so which they themselves may absorb the drug. So absorption of tetracycline capsules is determined by the presence of calcium phosphate, where calcium phosphate is used as diluent and forms insoluble complexes. Then compressed tablet, coated tablet. Compressed tablet, moist granulation is a common thing of the compressing of tablet. Binding agents used and adjuvants are used for the formation of granule. Disintegrant and lubricant of drug particle are poorly absorbed from GIT. Surface area for the absorption is also smaller. So viability of compressed tablet is also lesser than compared to the solution. Then comes coated tablet. So coating of tablet should be dissolved priorly before absorption. So time for this affect viability. Coating is used to make disagreeable drug palatable or to protect drug during the storage. So coating which we used are sugar coating, film coating and antric coating, which shows the variation or change in viability. So in future viability studies will be emphasized much more, and insistence on the studies conducted can be made by FD. Dosage form factors also include particle size, solvent, hydrate salts, polymorphic forms, complexes, pH, solubility characteristics, vehicle, manufacturing process, etc. Then manufacturing variable, the addition of various excipient in the process of manufacturing of dosage form and various processes like compression forces may affect the viability. So example now is addition of lubricant, which are generally hydrophobic in nature, which reduce wetting of the particle size, due to which the rate of dissolution is also reduced, and in turn the rate of viability of drug also get reduced. So excipient also interact with the drug and which also affect the viability. Then comes the dissolution rate. So for drugs which are poorly water soluble, dissolution rate of drug often rate limiting step, so viability of drug. Dissolution rate is determined by noise-Whitney equation, which is equal to dA upon H Cs minus Ch, where capital D is diffusion coefficient, A is surface area of drug, K is partition coefficient, H is thickness of stagnant layer. Then Cs stands for concentration of drug in saturated stagnant around drug particle, and Cv concentration of drug in bulk phase. Then physiological properties, that is gastric emptying time. Rapid absorption occurs if drug is given before meal, but certain irritant drugs like salicylates and iron salt are administered after the food to minimize GI irritation. Anticholinergic drug with prolonged gastric emptying time also decreases the absorption of drug. Increase in peristaltic movement reduces the drug absorption. GI transit time. The motility of stomach is important to the rate at which orally administered drug is passed on intestine. So delayed gastric emptying time will ultimately reduce the absorption of orally administered aspirin. So food also affects gastric emptying time. Absorption of amoxicillin, ampicillin and cefaxillin is reduced in presence of food. So this is due to the enhanced gastric emptying time. Then first pass metabolism. It means biotransformation of drug during absorption, through which intestine and the transport through the liver in portal circulation. So it is significantly reducing the percentage of oral dose and systemic circulation. Drug like propranolol, chlorpromazine significantly metabolized in liver first pass effect. When drug passes through the liver in first time, it is either destroyed in GIT or inactivated by liver. Then sublingual route of a drug administration can be used as alternative route. In this case, first pass metabolism is avoided, since drugs like isoprenaline and nitroglycerin, they are given sublingually, which directly enters the systemic circulation. Then comes disease state of gut. Absorption of drug may be affected by the certain condition like malabsorption, a chlorhydria, and cirrhosis of liver. Okay. Thank you very much for your patience listening. Thank you, friends.